We are very excited to have Sam Sober from Emory kicking things off. What do I do here?
Yep. It's a recent self-portrait. Cool.
Very excited to have Sam here to kick off our Monday meetings, following up with the CABI event on Friday that probably most of you were at as well. We're excited to see all the turnout for Mark Desposito's talk on Friday. So it's great that people are getting out and engaging in the broader community. Sam, as you see, an expert in finches. Sam did his undergrad work at Wesleyan University and then his PhD and postdoc at University of California, San Francisco. He's now an assistant professor over at Emory. Does fantastic work in multisensory integration, sensorimotor integration, especially in the context of vocal learning and the finches, as we're going to hear about. A great scientist, also. I'm fortunate to count him as a friend and excited to hear about what he's been up to here today. So thanks, Sam, for coming.
Okay, thank you very much for that nice intro. And yeah, it's great to be here. So the goal of our lab is to understand how the brain controls complex motor skills, and also how the brain uses sensory feedback to correct errors in behavior during sensorimotor learning. And these are, I think, really central questions for our understanding of complex brain function, because pretty much all of the complex skills that we rely on every day, these are all learned by a process of sensory feedback, both to initially acquire those skills and also to keep them maintained throughout adulthood. So to understand these general questions, we have some very, I think, interesting and complicated relationships that we have to get our heads around. So first we have to understand how networks of neurons in the brain control the pattern of muscle activity that we need to generate behavior. We need to understand the mechanics by which those muscles evoke the behavior of interest. And then I think maybe most interesting and most difficult is we have to understand how sensory feedback during behavior effectively rewires the brain to instantiate sensorimotor learning. And given the complexity of these relationships, our lab uses, as do other labs studying the same thing, a really wide range of different techniques to attack different aspects of the question. This is a very nice Dr. Seuss illustration that illustrates how I sometimes feel when working on manuscripts and grants. And in the course of this talk, I'll be touching on most, maybe not all of these things to talk about recent work that we've done. The first thing I'm going to start talking with, I'm going to begin with the behavior that we care about. So I'm going to talk about vocal learning in songbirds. And I'm going to talk about a series of ongoing experiments that use manipulations of sensory feedback and then quantitative analysis of behavior to try to understand the overall computations that are going on during a particular form of learning. So before I get to these experimental studies, I want to introduce the behavior that we study. So in the finches that we work with, young birds are not born knowing how to sing their songs. Rather, they copy their songs from an adult, usually the bird's father, though not necessarily. And then, so they memorize that song and then perfect the production of it by a process of trial and error learning. So I'm going to be showing a lot of these spectrographic plots, so I'll just briefly orient you to these. So this is a visual representation of an acoustic signal. So on the horizontal axis, we have time. On the vertical axis, we have the different acoustic frequencies of sound that can be produced. And the heat map here shows the power at each acoustic frequency as a function of time. So if we look up here at the song of an adult bird, you can see that somewhat similar to human speech, bird song is made up of these discrete vocalizations. We refer to these as song syllables separated by silence. So what I'm showing here, this is an adult song that is tutoring, which is to say teaching this younger bird how to sing. So birds begin to vocalize a little bit before 50 days after being hatched. And what you can see here is that when birds first begin to vocalize, they have this very quiet, sort of disorganized song that bears very little resemblance to the mature song of an adult. But this bird, after having just about 40 days of practice between here and here, has learned how to copy the song of the adult. So you'll see that this spectrogram looks almost exactly like that spectrogram. And there are many different ways of measuring the final success of song copying, but sort of no matter how you do it, you get 85, 90, 95%. And one of the things that's really amazing about this is that the continued presence of the tutor is not necessary for this kind of learning to occur. So you can expose this young bird to the song of the tutor, 10 or 20 times very early in life, remove the tutor, and then the young bird, through this process of trial and error learning, will learn how to produce the tutor's song. So what's going on here is that the young bird is comparing the sound of his own auditory feedback with some sort of target that's derived from this memorized version of the tutor's song. So this is a very simple diagram showing sort of the current thinking in the field. So the idea is that the way song acquisition happens is the young bird, the motor system drives song, and then the bird is comparing auditory feedback to this long-lived auditory target derived from the song of the tutor, computing the mismatch or error between those two signals, and using that error to update the motor system. And one of the other things I think is really incredible about this behavior is that after this initial and really rapid stage of early sensorimotor plasticity, a bird's song is incredibly stable throughout adulthood. So I could come back and record the song of this same bird 2-3 years later, and the song would be almost entirely unchanged. And when I first started studying birdsong as a postdoc, basically no one had really paid much attention to how it is that birds are able to maintain this incredible stereotypy of their songs throughout life. So the first set of experimental studies I'll tell you about were aimed at trying to understand how it is that after this incredible period of behavioral plasticity, birds are able to keep their songs on target. So our hypothesis here, and there's a couple of possibilities. So it could be that after this initial pattern of learning, initial period of learning, it could be that this error correction process just stops and the bird locks its motor, its motor state into some particular stage for the rest of his life. We knew going in that probably wasn't true. So in birds, as in humans, if a bird is deafened in adulthood, the motor program for song will fall apart over time. And something very similar happens in humans that go deaf later in life. So our hypothesis then was that this incredible stability of adult birdsong is based on a lifelong process of error correction, where birds are constantly monitoring their auditory feedback, detecting any small mistakes that they might be making, and using that error signal to drive a correction in behavior. So to test this hypothesis, we had to come up with a way to introduce a correctable auditory error in sensory feedback that a bird was receiving. So we basically needed to figure out a way to trick a bird into thinking he was making a mistake, and then he could then correct. And this is what we came up with. So this is a Bengalese finch wearing a set of miniaturized headphones. I stay in science. You can dedicate 2 years of your postdoc life to figuring out how to make really little headphones. So the way the system works is that the bird sings, there's a microphone in the cage. The acoustic signal is routed through online sound processing hardware that lets us introduce arbitrary manipulations of the acoustic signal, which are then played back through the speakers in the headphones. And this whole system has a loop delay of about 10 milliseconds, and in various supplemental figures we've shown that delay by itself does not introduce any detectable change in song. Okay, so just to remind you, the hypothesis is that adult songbirds are maintaining their songs by a process of error correction. And so our specific hypothesis for this experiment is that in an adult bird, if we trick him into thinking that he's singing one semitone too high, for example, then the bird should drop the pitch of his song to correct for that, and vice versa. Yeah, sorry, and I should mention, so the first experiments I'll tell you about involve these one semitone manipulations. Musicians will know that one semitone is the same pitch interval between two consecutive keys on adjacent keys on a piano. It's about a 6% change in absolute frequency. Okay, so I'll show you a representative experiment now. So we would put the headphones on the bird, we would let them get used to wearing them. Then we would introduce the pitch shift in a single step. So this first study, we're going to increase the pitch of auditory feedback to test whether the bird is going to drop the pitch of his song. And the way that we monitor changes in vocal output over time is by repeatedly measuring the pitch of different song syllables. So this is a spectrogram from this bird, and I'm showing the 4 syllables that we're quantifying. And we're going to measure the extent to which the pitch of each of these guys change over time. So what I'm showing here, each of these the 4 colored traces here show changes in the pitch of these particular syllables. The black line is the average of those, so I'm going to use the black line to be a measure of change in pitch of song over time. Okay, so at the beginning we increased the pitch of auditory feedback. You can see that over the next 14 days the bird is gradually dropping the pitch of his song. One thing I want to point out is that the pitch shifts that we're introducing are always relative to the bird's current vocal production. So what that means is that as the bird, so we're always shifting the pitch upward by 1 semitone. So as the bird begins to drop the pitch of his song, the experienced auditory error is getting closer and closer to 0. So this is very important. This is a correctable perturbation that we're giving. So had the bird dropped his pitch by 1 semitone, there would have been 0 auditory error. One thing you'll notice is that this error correction is incomplete. It stops about halfway, and in longer experiments where we let them stay at asymptote for a while, it usually stays around halfway. I'll have a lot more to say about why that is in a minute. Then after the 14th shift day, we set the pitch shift back to 0, and you can see that there's the beginning of a return to baseline. And we come back and look much later, you see that this return to baseline is almost complete. So 2 things about this. This is, one experiment, obviously, but this kind of thing shows, first of all, I think it's very strong evidence that birds really are constantly performing this process of error correction in adulthood. And one of the things I really like about these experiments is that this finding that pitch comes back to baseline is incredible. It happens in every single bird. So this is also really strong evidence that birds have this lifelong target that they're constantly seeking to get their pitch to, and even after a couple of months of us putting headphones on and doing weird stuff like that, they're still able to maintain both the target and the ability to get their songs there. This is a reminder for me to tell you that this is symmetrical. So this was an example. We increase the pitch of auditory feedback. Birds lower the pitch of their song. Works the other way too. We drop the pitch of auditory feedback. Birds increase the pitch of their song. So this is average data across a bunch of birds. So you can see that in response to a 1 semitone pitch shift, like what I just showed you in the example, birds correct about 40% of that imposed shift. And as I said, I think this is very strong evidence that the stability of adult song does not result from sort of locking the motor system into some dynamical state or some set of parameters, but rather through this lifelong process of error correction. So this first demonstration of adult learning was during my postdoc, and then since then we've put out a series of papers trying to answer this question of how much does the brain compute how much to change behavior based on some sensory error. And this, I think, is also a really, really fundamental question in sensorimotor learning. And the reason that this is a hard problem for the brain is that the brain is kind of stuck in this dilemma, which is that for complex behaviors like this, the brain has to rely on sensory feedback to keep the behavior calibrated. But the problem is that sensory feedback is inherently unreliable. So there's noise in the environment, and there's noise in sensory encoders, and more centrally as well, probably. So for some piece of sensory information that tells the brain that an error has occurred, how does the brain compute how much it should change behavior? And I'll be telling you about this now. So this first thing we did, the data I already showed you rather, is that for a 1 semitone shift, birds correct about 40% of it. So the next thing we did was a very simple experiment. We just varied the magnitude of the sensory error. And what you can see was this pattern that was initially very surprising to us. So for a 1 semitone error, that's the blue trace as you've seen. Next thing we did was we dropped the size of the error to half a semitone. And somewhat surprising to us at first was that learning actually got significantly faster. So you can see that this trace pops up faster. And it's also significantly more complete. And by that I mean that in response to a 1 semitone error, the bird's correcting about 40% of it. That fraction goes up to about 70% with a smaller error. And then in the other direction, when we used a larger error, 1.5 semitone, you can see from the green trace that the learning is again getting slower with a larger error. It's also less complete. And when we crank the error up to 3 semitones, learning approaches 0. So we have then an inverse relationship between the magnitude of the sensory error and both the speed and completeness of sensorimotor learning. So what this tells us then is that this kind of simple-minded cartoon of sensorimotor learning I showed you a little while ago, this isn't right. In fact, it's almost exactly wrong. So remember, this model predicts that the bigger the difference between auditory feedback and auditory target, the bigger the error signal, the more change in learning. In fact, over this range of parameters, exactly the opposite thing is happening. The bigger the sensory error, the slower and the less complete the learning is. So here we've got, in the same population of adult songbirds, smaller errors give faster and better learning. A second set of experiments we did was looking at younger birds versus older birds. So these are, when I say young adults, I mean these are birds who have just barely learned to sing. This is 90 or 100-day-old birds versus the adults, which is the popular, these guys are about 100 days old to a year or so, which are the population I showed you here. So here what we're doing is we're giving the same pitch shift, a 1 semitone pitch shift, to younger birds versus older birds. And you can see here, excuse me, you can see here that for the same sensory error, learning is significantly faster and significantly larger for the younger animals. So what's going on here? We've got this case of exactly the opposite pattern you'd predict from that sort of cartoon that I showed you earlier, and the same sensory error giving these very different dynamics of learning in younger versus older animals. Okay, so why should this be? So the first thing we started thinking about was that we had this intuition that we could explain this pattern of behavior by thinking about the probability distribution of auditory feedback. In this case, the probability distribution of pitches that birds hear before we start doing these manipulations. So even in adult songbirds that have fully learned their songs, there's substantial variation, trial by trial variation in the pitch of particular song syllables. So this is one song syllable from our data set, and this is the distribution of pitches. Here what I'm doing is I've replaced that example distribution with a normal distribution of pitches where the width of this distribution is matched to the average standard deviation in our data set. And I want you to think of this as, so think of this as a probability distribution of how the bird is accustomed to hearing himself sing. So what I'm showing now is this red trace is just this blue trace shifted to the right by half a semitone. And remember the 0.5 semitones is the smallest error that we tested that I showed you before. This introduces learning that is relatively fast and relatively complete. Now what I'm showing is for the 1.5 semitone, and what I should mention here, I forgot to mention, is that this gray shaded area shows that there's a lot of overlap between these probability distributions. And what that means then is that once the bird starts experiencing the half semitone shift, most of the pitches that he hears himself sing could have come from what I'm going to call his prior distribution. So he's got this prior on what his song is supposed to sound like. And for these small pitch shifts, most of what he hears himself sing through the headphones could have come from the prior. What you can see here is that for half semitone and 3 semitone shifts, the shift gets bigger, learning gets slower and less complete, and the area of overlap here gets smaller and smaller. And what you can see is, remember, 3 semitone shifts, that's where learning basically falls to 0. It also happens to be where there's essentially no overlap between the bird's prior distribution and the sensory information that he's getting there. So this, it's by no means a full model, but it gave us a clue that we could think about. We could maybe try to figure out what's going on with these different patterns of learning by thinking about the overlap between the sensory evidence that's coming in and the bird's prior. Using no additional parameters, that same principle or heuristic or whatever you want to call it can also explain this age-dependent change in learning. And I should mention, as many of you may be thinking, there are many, many, many papers out there that show that younger animals learn faster than older animals. We've all experienced this. But as far as, but what's new here is that it turns out that you can predict essentially all of this difference, all of the difference in learning speed and efficiency between the younger and the older guys, but with this principle of overlap between the prior and the sensory feedback. So this is illustrated here. I showed you these 2 panels already. So for an older adult, small shift, lots of overlap, lots of learning. Bigger shift, less overlap, less learning. But for the same size shift here, you can see that younger animals are more variable. This is another thing that is true not just in songbirds, but in sort of all sorts of behavior. Younger animals, they learn faster and they're a lot more variable. So what's shown here is you'll notice that the prior for the younger bird is about twice as wide as the prior for the older bird. And what that means is that for the same shift of the probability distribution, there's a much greater fraction of overlap here. And again, this difference in overlap, for the same error but a different prior distribution, can fully explain the difference between these patterns. Okay, so this overlap principle, it can predict the learning outcomes. We can do a pretty good job of predicting, if we're just thinking about learning as this exponential approach to some sort of asymptote. We can do a good job of predicting the parameters of that model just by looking at this overlap. But that's not really what we want to know. What we want to know are the dynamics of, for a particular piece of error information, how is it that the bird is updating his motor strategy based on both his prior history of production and also the new sensory information. So now I'll tell you about some, this is work in progress that we're doing with Ilya Nememin. He's in Emory Biology and Physics, and his graduate student Baohua Zhou, who's in the physics department. And we're using the framework of Bayesian inference to think about how this new evidence is used to update a motor policy. So many of you are probably already familiar with this, but just very briefly. So Bayesian inference gives us a way of thinking, for some prior distribution. So, in this case, think of it as the bird's sort of selection of motor commands of how he sings a particular syllable. What Bayesian inference does is it tells us how we should update that prior based on some evidence. And the form of this new evidence we're going to call likelihood. This is a correction to the prior that's suggested by new data. And in this case, the new data is the bird's auditory feedback of his own song. And in Bayesian inference, you multiply these things together, and then proportional to that, you get a change in the posterior. So the posterior is the updated version of the prior given the new evidence. So the way we're modeling this now is, as I've said qualitatively, we've got some prior distribution on motor commands. Each time the bird sings, he draws a sample from that. The bird then observes some sensory consequence, and by sensory consequence I mean the sound that reaches his ears. And this is where we can introduce our manipulation by shifting the pitch that comes through the headphones. Then, so let's use this as an example. So let's say, for his whole life until I put headphones on him, the bird is happily singing along with this prior. It's working pretty well for him. Then what I do is I put the headphones on, and let's imagine that I'm downshifting the pitch. So he's hearing himself singing too low. The likelihood function here is that he should probably be singing higher given the sensory input that he's getting. So the likelihood would be up here. And then from Bayesian inference, the idea of how to update the posterior should be somewhere in the middle. It should be the likelihood and the prior multiplied together. And that's what's shown here. And this is what makes this an iterative model, a Bayesian inference model, is the idea is that the posterior is then used to update the prior. So the idea here is we've got, based on each successive waves of incoming information, the bird is continually updating his prior distribution. And this, if this works, will give us a Bayesian model of how the bird is changing his behavior throughout time. I think that's it for equations. No, sorry, that's not it for equations for the rest of the talk. Sorry. Okay, so how do we actually implement this? So let's think about where these things would actually come from for a bird. So what about the sensor? What about the likelihood? What about the sensory feedback that the bird is receiving? So one thing that's important to keep in mind, and I should have mentioned this, my PhD work was on how humans combine different streams of sensory information. So specifically, I was looking at arm movements. This was how people combine visual and proprioceptive information from the arm. And this was sort of very much in our minds as we were working on this. And it turns out that my old stuff and other people's stuff as well has shown that the brain is in fact incredibly flexible in how it chooses to rely on different sensory modalities. So my dissertation work, for example, showed that if you change something about the task, like whether you're reaching to a visual target or trying to reach to your other hand, the brain can really rapidly switch from relying on visual feedback to relying on proprioceptive feedback. So one of the things this means is that we should always keep in mind that auditory feedback is very powerful for remapping vocal behavior, but it's not the only sensory modality that you rely on when you're controlling your voice. So for example, we have proprioception throughout the vocal tract. We also probably have internal models that tell us, given a particular motor command, what should our sensory feedback be. So the idea here is that when the bird is wearing headphones and is receiving this pitch-shifted auditory feedback, he's really got multiple sources of sensory information. He'll have the auditory version, which is manipulated however we're manipulating it, and then he's got some number of unmanipulated sensory modalities, and this could include proprioception or forward model predictions, etc. So how do you combine these things into a likelihood? You've got all of these different kinds of sensory information. How do you get the likelihood function? Now, the way the Bayes optimal way, and this is the way that most people assume this happens, is that you just multiply all of these distributions together and to get a likelihood function. So you're just multiplying normal distributions, you get a normal distribution. And then that could serve as your likelihood function, and you get your posterior somewhere in the middle. Here I'm drawing all these various distributions as having the same variance. So in this case, if you've got two normal distributions, you multiply them together, what you get is a normal distribution right in the middle. There's a problem with this though, which is that if this is the way that the songbird brain is combining these different streams of sensory information, the bigger the shift in auditory feedback, the farther out the likelihood function is going to be. So the further this moves over, the further this is going to move over. And because the posterior is always some fixed proportion, in this case halfway in between the likelihood and the prior, what this means is that the farther we shift auditory feedback, the farther the posterior is going to go, the more learning is going to happen. But we know that's the opposite of what happens. In fact, the farther we push auditory feedback, the learning eventually goes away, it gets smaller. So it turns out that you can, so this does a bad model of this does not predict the data. What does predict the data is if we relax this assumption that the likelihood function is a multiplicative function of these various sensory inputs. So here, there are a few ways to do this. So if instead of multiplying these together, we just added them and normalized, we would get a two-peaked function. Also, if we thought of these not as normal distributions, but as something that has an offset, we could multiply them together and get a two-peaked function with some tails. So one of the things that is what's important about this is, so let's imagine that we now have a two-peaked likelihood function. And furthermore, let's say we've got that two-peaked likelihood function for a really big pitch shift. That's what I'm showing down here. What you'll see is that this peak of the likelihood function gets multiplied by 0. It does not affect the posterior at all. This one is centered at 0 because it's the peak that results from the non-shifted feedback, and learning won't happen. So this is a way that gives us this disappearance of learning for large error sizes. What I'm not showing here is that for really small shifts of the auditory peak, this is basically a one-peak likelihood function, and you get pretty robust learning. So what this shows then is that by sort of tweaking this assumption of most Bayesian models, which is to say this idea that you only ever have a single peak likelihood function, we can capture all of the data that I just showed you. But that's not that surprising, because we sat around scratching our heads trying to come up with a model that would fit our data. And we did it by introducing this change. But of course what you really want out of a model is for it to make surprising predictions that you did not build into what you're creating, and that's what we did, or that's what we found. So actually, let me back up. So remember from the formulation of this that the posterior, the posterior then propagates into the prior. So the idea is that as we start manipulating the shape of the bird's likelihood function, eventually that should change the shape of the prior, of his distribution of motor commands. And one of the things, and this is data that I'll show you. This is from a slightly different experiment from the ones I was showing you before. So whereas previously we would just introduce a single error, here what we're doing is we're introducing a staircase of errors. So every few days we increase the size of the pitch shift a little bit more. So what's nice about this is it drives really strong learning. So here we're driving more than one semitone of pitch change. So one of the things that, and this was not built into the model, is that the model predicts that the variability of the distribution of pitches should increase monotonically over time. And that in fact happens. And the reason that variability should increase over time is, remember that if you have a two-peak likelihood function and that propagates into the prior eventually, you're eventually going to get a double-peaked prior distribution. And that in fact is what happens. So this is, again, this is we've got the model predictions in red here and the data are in blue. What this shows is that for some subset of gestures, you start to build up this other, this second peak in the pitch distribution. And the peak sizes here don't match yet. We're working on it. But what this really suggests then is that the way that the bird is combining these different sources of sensory information is leading to these very unusual and unusual distributions of the motor command and things that sort of contradict the kind of the typical Bayesian learning models. Okay, so I've just told you about some of the behavioral studies. So this is sort of a pitch for what you can do just with behavior and computation. What I'm going to tell you about next is some work that we're doing, sort of trying to looking inside the skull, trying to figure out what are the different neural systems that are involved in controlling this kind of sensorimotor learning. So in addition to looking charming in headphones and doing this really computationally interesting behavior, the songbird, the different neural pathways in the songbirds, I think, are one of the things that make it such a great model system. And I'll summarize many decades of work by many different labs with this picture here. So one of the things that's terrific about the songbird brain is that there's this spatial separation of the neural circuits that are required for the production of the behavior and for the learning of the behavior. So don't worry about what these acronyms mean, but you've got some of these nuclei that are motor areas. So if you lesion them at any time during the bird's life, he won't be able to produce the song. And those are spatially separated from these areas I'm showing in green, which are required for learning. So you damage these at any time during the bird's life. The bird's vocal production won't change, but he won't be able to learn anything new. So you lesion it early in life, the bird never learns how to copy the tutor's song. You lesion it later in life, and certain forms of vocal learning can no longer take place. But one of the things that has been, and people discovered this, decades ago from a lot of very nice lesion experiments. And, so there's this idea that, somewhere in this circuit, probably in the green part of the circuit, that must be where the error signals are. So these parts of the brain that if you lesion them, the bird can't learn, that must be where the neurons are processing sensory errors. Then over the last maybe 5 or 8 years, as people have been doing neural recordings in behaving songbirds that were experiencing errors in auditory feedback, there's this really surprising finding that's been replicated now in 5 or 6 papers, which is that there's no spiking response as a result of auditory errors anywhere. Not anywhere in the brain. At the cochlea, of course, there is, but not in any of these parts of the song pathway that are required for vocal learning. So what's going on here? Why is there, if this is the error circuit, where's the error signal if we can't record it when we stick our electrodes in? And there's been an idea that this has been hypothesized for a really long time, but I think not tested until we did the work that I'm about to tell you about, which is that the error signal could be carried by dopaminergic neurons in the midbrain that project into the basal ganglia. So I promise no acronyms, but I'm going to lie. This area that is called area X is a subnucleus of the songbird basal ganglia that is only involved in as far as anyone can tell, is only involved in vocal learning. The reason it's called area X is embarrassing. People didn't know what it was for a while, so they called it area X. There's actually nothing worse than songbird nomenclature. Okay, so like the rest of the basal ganglia, both in songbirds and in mammals, there's massive dopaminergic input there. So I told you that none of these classic song system, what we think of as the classical song system nuclei, have any response to auditory errors. However, before we started, we knew there were dopamine inputs to X, and we also knew that some of the inputs to VTA, S, and C, which is what this dopaminergic part of the brain is called, some of these neurons are responsive to auditory errors. So our hypothesis then was that the way error information is getting in to the song system, to this network of nuclei, is via these dopaminergic neurons. Oh, sorry, right. So what I'm showing here, this is a sagittal section of a songbird brain. So anterior's over here, dorsal's up there. This is stained for tyrosine hydroxylase. So the darker the brown, more or less, approximately speaking, the darker the brown, the more dopamine there is there. So you can see these are the cell bodies in the VTA, S, and C. These blue lines I drew. That's not really what the brain looks like, although our lives would be much easier if it did. And you can see that these dopamine neurons are known to ramify throughout the basal ganglia. And it's a little hard to see on this plot, but will hopefully be easier in a minute. Area X stains a little bit darker for tyrosine hydroxylase. So that's this part of the brain. The fluorescence doesn't show that well. This is just neuroanatomy confirming that there are, in fact, in the species that we study, that there are, in fact, these X-projecting dopamine neurons. Okay, so to test this hypothesis that these are the neurons that are providing the error signal to the motor system, we're doing an experiment where we're going to selectively lesion the dopamine inputs to area X and then see whether that affects the bird's vocal performance and vocal learning. So we take a neurotoxic approach. We do these very localized injections of this compound called 6-OHDA, which kill the dopamine neurons that project to area X, but spare the dopamine neurons that project to other parts of the basal ganglia. To measure learning, in the published paper, we did not use headphones. We used this other learning paradigm that I'll tell you about now. So this is a form of learning that drives the same kind of change in behavior as headphones. We can drive a change in pitch in how the bird sings. But the way we do it is a bit different. So we have a software that constantly monitors the bird's vocal production. The software can detect when the bird is singing a particular syllable, and then in about 8 milliseconds compute the pitch of that syllable. What we do then is we set up a conditionality like this, where if the pitch is above or below some threshold, which I'm showing with this dashed line here, we'll either play a blast of white noise through a speaker to punish the bird for producing that sound, or we will let him escape. We won't play any blast of white noise. Birds hate blasts of white noise. And what happens, so this is an example experiment where we're hitting the bird with white noise if he sings a lower pitch. We're allowing him to avoid that white noise if he sings a higher pitch. And this manipulation will very robustly drive, in this case, an increase in pitch over time. This is just a spectrogram showing, this would be the targeted syllable here. He's not getting hit with white noise there. But this very bright band of acoustic power, that's the psht sound coming out of the speaker. Okay. So the way we run these experiments is we first do one of these. So we quantify the bird's ability to learn over about 3 days, 3 days of white noise exposure. Then we immediately lesion the dopamine inputs to this part of the basal ganglia. About a week of recovery, and then we retest on the same animal. This is just, I'm going to skip this unless people really want to know about it. This is just showing, so you can see, I hope, that in a sham-treated animal, we've got dark staining for the dopamine enzyme here. Gets much lighter when there's a lesion. And by various measures, we're knocking down dopamine by about 25 to 50%. So this is not a complete lesion of dopamine inputs. But importantly, we're not killing the cell bodies. So we have figured out how to selectively kill the dopamine fibers, but not the neurons that live there. Okay, so this would be pre-lesion. Oops, that would be the wrong way, sorry. So then in the same bird, a week after doing the surgery, we retest them. You can see that, that there is some residual learning, but the magnitude of learning has gone way down. This is averaged across all the birds in our sample. So this is pre-lesion and post-lesion animals and you can see that when retested, the magnitude of learning is falling by about half. We don't see any significant change in learning for sham-treated animals that just have saline injected. So what this tells us then is that this particular form of vocal learning, I'm going to call it vocal reinforcement learning, because with the headphones, the bird is he's evaluating his own sensory feedback without any external punishment or positive reinforcement from the experimenter. Here, it's really artificial negative reinforcement being provided by the experimenter. So I'm going to call this reinforcement learning. And you see this very clear dependence on an intact dopamine system. Here's something really interesting. So at the end of these white noise experiments, we've driven a pretty big change in vocal pitch. And then what we do is after the third white noise day, we just let him sing for a few days. So we remove this artificial negative reinforcer. And what that means is that on the first on the first day after white noise turns off, so let's look at the pre-lesion group first. So thanks to this reinforcement, he's changed his pitch a lot. This is about 0.6 semitones. Then what happens is he performs this, the bird will perform this process of sensorimotor error correction that's completely internally guided. It's not being manipulated by us. And you can see this exponential decay back to baseline, just like we saw these exponential learning curves when we were manipulating auditory feedback via the headphones. With the white noise learning, there's obviously this huge deficit in following dopamine lesion. Absolutely no deficit in the recovery. In fact, it's a little faster in the recovery, which we would actually expect from the results of the headphones experiments. Remember, you're starting off with a smaller error. We've already seen smaller errors are learned faster. So we see then what seems to be the case, and this is something that we're following up now, is that there's this, even though the final output of these forms of learning is the same, it's the bird learning to change the pitch of his song syllables, depending on the structure of the task, whether it's an external reinforcer or the bird's self-evaluation, there seems to be this differential element of dopamine. And we're following this up not only by doing headphones type sensorimotor learning experiments in dopamine lesioned animals, but also by doing neural recordings from the basal ganglia while the birds are performing this task with and without dopamine lesions. Sorry, I completely forgot. Break in any time with questions if you have them. I didn't want any questions up till now, but now I'm willing to take it. No, I'm just kidding. Okay. So what was that? No.
Is the lesioning permanent?
That's a good question. So there is so we haven't checked yet. So 6-OHDA is used pretty commonly in mammalian experiments. Sometimes there can be regrowth after 6-OHDA. I mean, what we do is immediately at the end of the experiment, we process the tissue. So we haven't looked, but it's possible that there is. Anything else? Yeah.
So if I remember correctly, you got generally a larger signal when you have the white noise, a larger change, adaptive change, the white noise versus the pitch change.
Yep.
Why do you think that is?
It's a great question. So when you start studying dopamine, you start to think that everything's about dopamine. I don't know if you knew this, but let's pretend it's super simple and dopamine is just your error knob. Maybe getting blasted with white noise cranks the error knob up more. Maybe this makes me think that's not true, though. I mean, the fact that you can have a really dramatic effect of dopamine lesion on one kind of vocal pitch learning, but not another one, makes me think that that's wrong. My guess, and this is total speculation at this point, is that there's two kinds of the mechanisms of learning up until you get to the bits of the motor system that are controlling pitch are completely different for a bird's self-evaluation and how the environment is reacting to the bird. I mean, so a bird, he's using a song for courtship. Either the female's interested or she's not. There's this kind of binary outcome. Birds never get blasted with white noise in the wild throughout evolution. So I'm not saying that it's a naturalistic punishment.
But they might isn't interested or not.
Yeah, exactly. So if I had to guess, it's because the computational structure and the neural implementation before you get to area X is different for self-guided error correction versus the environment punishing you for what you did. That's a guess. Okay. Okay, so I've just told you about this dopamine project involves manipulating sensory feedback while doing these different neural manipulations and looking at the consequences for behavior. The last set of studies I'll tell you about are really focused on motor coordination and how neurons encode motor output and take advantage of the biomechanical properties of muscles. So this is an approach that, as you'll see, involves looking at the transformation of signals from neurons to muscles and then from muscles to behavior. So I want to take a little step back here and just talk about different theories of motor coding in the cortex. So we all believe that behavior is controlled by patterns of neural activity in the motor system, and lots and lots of work in neuroscience, our lab certainly included, are trying to understand the relationship between a spike train that you record in the motor system and the behavior that results from it. And there are, very broadly speaking, two different ways of thinking about this. So what I'm showing here, this is a hypothetical neuron, and this would be on 6 different recordings from the same neuron on 6 different trials of the animal doing some behavior. And as we've talked about before, all behavior is variable, so no animal ever does exactly the same thing from trial to trial. So if we're trying to understand how patterns of spiking activity drive behavior, one possibility is that what matters is spike rate. So in these 6 trials, I'm showing 3 of them with a low firing rate and 3 of them with a higher firing rate. And in fact, a lot of the sort of classic early work in motor system was looking at rate coding. So the idea is that what really matters is over some time window, how many spikes is this neuron firing for shaping the motor output. Alternately, it could be that the information, or the signal that's actually driving behavior, is not about spike rate, but is rather about the precise timing of spikes. So here for the same imaginary neuron, or a different imaginary neuron, same number of spikes, it's always 4 spikes, but you can see that the particular timing patterns are different. And so if hypothetically, the neuron, if there were a timing code, you might expect to see a different behavioral output on trials 1 through 3 versus 4 through 6. Now many of you know this, I'm sure, that there's a really long history and really big literature about spike timing codes in sensory systems. So for whichever sensory system you can think of, there have been some papers demonstrating that you can learn more about the sensory stimulus if you take spike timing into account than if you're just looking at a spike rate. But one of the things that I think has always been a real problem with that literature is it's very hard to figure out whether or how different, a millisecond difference in spike timing actually affects anything downstream, whether it's downstream, activity in downstream neural areas or perception or behavior. So what we want to know here is if we look in motor cortex, how much does spike timing matter for predicting and controlling the behavior compared to spike rate? So I've already told you, song is composed of syllables. There's measurable trial-by-trial variation in the pitch of a particular song syllable. The area of the brain that is, we think of this as analogous to vocal motor cortex, is this area called RA. It's analogous. I think it's analogous to layer 5 of motor cortex. And the reason for that is that all of the projection neurons go directly to the brainstem motor neurons, which then directly innervate the vocal muscles. And we know from lots of lesion and stimulation studies that neurons in RA are intimately involved in shaping the acoustics of vocal production. So when we go in and record neurons in area RA, so it looks like this. So what I'm showing is this is the same neuron being recorded on 4 different iterations when the bird is singing this sequence of syllables. So what you can see, a couple of things you can see here is RA neurons fire bursts of action potentials. These action potentials happen at very stereotyped times across trials. In fact, here it looks somewhat like they're locked to the onsets of syllables. So it's very reliable that this neuron is going to fire a burst at these 5 times here. Another thing you can see is that if you look across trials, you can see that there's variability in both spike count and spike timing. So for the syllables for the spikes that immediately precede this song syllable, sometimes it fires 1 spike, sometimes 2, sometimes 4. Another thing that you have to look a little bit harder to is that there's also variation in the precise timing interspike interval pattern sequences of this neuron. So this burst and this burst, they both contain the same number of spikes, I think 7. But you can see that the precise interspike interval pattern is different. So what we want to do then is, the first step in this project was to quantify how much we can learn about vocal output. In this case, we want to know, can we predict where the pitch of this syllable is going to fall from looking at both rate and timing? And so the way we do this is we take, for some syllable, we'll take the full distribution of pitches. We'll just divide, we're just going to split it in half into the lower pitched half and the higher pitched half of renditions. Sorry. I should back up here. So pitch, we pick some convenient time during the song syllable to measure pitch. So in this case, this would be, 10 milliseconds after the onset of the syllable. And then we record as many spike trains as we can from single neurons. So this is something, this is of course our best neuron, or it wouldn't be on the slide. This is, a few hundred spike trains recorded during the production of this song syllable. Then we want to ask how informative are rate and timing in predicting whether a particular iteration of the syllable will be lower or higher pitched. So the way we do this is by computing a term we call mutual information. So what we want to know is how much does observing the pattern of spikes from this neuron, how much does this reduce our uncertainty about whether that syllable is of higher or lower pitch? And these types of quantities and analysis, this has been used really extensively in the sensory field, with minor adaptations using similar tools in the motor domain. So the way we do this is, what we want to know is the mutual information at different time resolutions. So imagine that we're looking at a 40 millisecond long spike train that happens right before the bird sings a syllable. We want to know how predictive that is about pitch. So what we can do is we can represent each of these, and these will be two hypothetical spike trains from a neuron. We want to represent these with different temporal resolutions. So let's say, for example, that we're just looking at spike count. So we're just counting the total number of spikes that happen in this 40 millisecond spike train. You'll see here that for these two spike trains at 40 millisecond resolution, so just looking at spike count, we would represent them by the same word, 4 and 4. But as we go down to finer and finer temporal resolutions, you can see that eventually we get different representations or different words corresponding to each spike train. So the red one versus the blue one here. And what this means then is, so let's say, for example, that this spike timing pattern were associated with a lower pitched rendition of a syllable, and this spike pattern were represented by a higher pitched rendition of a syllable. If we were just quantifying, if we were just looking at spike count, or just looking at a single 40 millisecond time bin, it's the same word here, so there can't be any information or any reduction of uncertainty about what the bird is singing just from looking at those words. On the other hand, when we go down to 5 millisecond resolutions, there'd be different words representing these two spike patterns, so there'd be greater than 0 information. We would learn something about what the bird were singing when we went down to 5 milliseconds. And the reason people go to the trouble of doing this is that this kind of analysis can tell you at what time scale is a neuron encoding something. Previous work was almost entirely focused about what a neuron was encoding about a sensory stimulus in the past. What we're doing is asking on what time scale does the spike train encode upcoming behavior. I won't go into this in detail. For mutual information aficionados in the house, this is the equation, and the hard part of doing this kind of computation is how do you compute these different entropy terms. We use the Nememan-Shafi, the NSB algorithm, because it's the best and because we were collaborating with Ilya Nememan. Okay, so here's what we find. So remember, we're looking at, we're representing the spike train at different resolutions. So when we just look at, and what we're trying to do is predict whether a syllable is at high or low pitch. So sort of surprisingly to us, when we looked at 40 millisecond resolution, which is to say, just counting up spikes, there's very, very little information about what the bird is about to sing. And if this neuron were using purely a rate code, so if it were only the total number of spikes in 40 milliseconds that are being used to control vocal pitch, we would expect to not learn anything else about the bird's upcoming behavior as we went to finer and finer temporal resolution. So we'd expect to see this. What we actually saw was this. We saw this huge increase in the amount that we could learn about what the bird was about to sing once we started breaking the spike trains down into 2 millisecond resolution or 1 millisecond resolution. And so what this then tells us is that you can do much better in predicting behavior from looking at fine patterns of spiking activity. It also, the corollary of this is that if we used a rate-based analysis, just looking at spike count, we would miss something like 90% of the information that was in the neural code. And this, I think, is this really, really struck me. This is in my own previous motor control work, all I was looking at was rate coding because that's sort of what everybody does. But at least in this context, in this analysis, I think there's strong evidence that you're going to miss most of what's going on if you're just counting spikes. So actually for this whole talk, I've just been talking about pitch because happens to be the acoustic parameter that we study in our behavioral experiments. Of course, there are lots of other parameters in acoustic stuff as well. We see basically the same results when we look at other acoustic properties too. Okay, so moving on to some more recent and ongoing stuff. So what I've just shown you is evidence that spike timing is informative, which means that if you look at spike timing, you can predict something about behavior. That's very, very different from a demonstration of causality. So what I've shown you so far does not, it's a tantalizing clue that the bird might be using these spike timing variations to control behavior, but it really doesn't show that these spike timing variations are actually physically causing the bird to produce different motor output. So a good chunk of the work in the lab now is focused at doing that, going from correlation to causation. So can we evaluate this hypothesis that not only are these little timing differences predictive, but they're actually controlling something. So here's how we think about it. So remember, all of that spike train analysis I was showing you was looking at area RA. Area RA does not control the behavior directly. Remember, RA projects to the motor neurons project to the muscles, and then the muscles control the behavior. So if, in fact, spike timing in the motor system is being used to control the behavior, I think that at least two things have to be true. First is that there has to be a spike timing code at the level of the motor neurons and at the level of the muscles. So our 2016 paper demonstrated that there's plenty of information in spike timing in RA, but maybe that spike timing information is just thrown away when you get to the brainstem and to the muscles, and it doesn't do anything. So that has to be true. The second thing is, okay, so let's say that there's a beautiful spike timing code in the motor neurons and in the muscle fibers themselves. That's not going to mean anything for motor control if the muscle, the force-producing properties of the muscles and also the physical properties of whatever the motor organ is can't transduce that. So these, I think, are, we've got parallel projects working on both of these, and I'll tell you about some of the early results now. So, okay, so I've been talking about muscles. So what do vocal muscles look like in a songbird? So this is a cartoon showing the syrinx. This is the vocal organ in a songbird. So you can see this is a more detailed cartoon version. So we've got the trachea that goes up to the beak and upper vocal tract. We've got the bronchi down here that go down to the lungs. These colored structures are some of the vocal muscles that surround the syrinx. So the contraction of these muscles actually shapes the acoustics coming out of the bird's beak. These muscles are extremely small and hard to work with, which is a real pain. Interestingly, they have extremely fast force dynamics. So from a mammalian striated muscle, like if somebody were to shock my bicep and measure the force output, the peak force would arrive in about 30 or 40 milliseconds after stimulation. So that's the twitch speed of typical mammalian striated muscle. That force peak in a songbird muscle would be reached at about 4 milliseconds. So these are extremely fast muscles that are capable of transducing very small differences in timing input. So as our way of measuring, of asking whether there is a spike timing code at the motor periphery, what we've done, and I'll show you how we've done this in a moment, is we developed techniques to record single unit EMG from behaving animals. So what do I mean by single unit EMG? So a motor unit is the basic unit of motor output. A motor unit consists of one motor neuron and the multiple muscle fibers that it innervates. So what happens is the motor neuron fires an action potential in a one-to-one and very precise way. This action potential causes an action potential to be fired almost perfectly synchronously in all of these muscle fibers. And if you set up your recording techniques just so, you can record the synchronous volley of this motor unit. So it looks kind of like a spike recorded from a neuron, but in fact it's a spike recorded from a single motor unit. So in collaboration with a company, we've devised this new kind of EMG array, which is this very, very miniaturized, flexible EMG thing that you suture it onto the muscles using these suture holes. The current version has 16 contacts. We can get these contacts down to 50 micron spacing. It actually works a little bit better at slightly larger spacing. And just to give you an idea, this muscle is a few millimeters long. These 16 green dots show the location of these 16 electrode contacts on that muscle after it's attached. When everything is working absolutely perfectly, you get signals that look like this. So this looks like a spike train recorded from a neuron. It's actually a spike train recorded from a muscle. You can see a zoom there. And for time reasons, I'll go through this fairly quickly. But in a couple of different parallel projects, we're recording. So to ask whether there is in fact a spike timing code at the motor periphery, we're recording spike trains during behavior both during singing. So this raster plot shows firing of a single motor unit, actually from this muscle on the syrinx here, as the bird sings. We're also looking at the control of respiratory air pressure. So all vocal behaviors, including speech and birdsong, really depend on the bird being able to control air pressure. So here, the behavior that we're measuring while the bird is singing is the acoustic output. What's being shown here, this is actually just during breathing, this is the rise and fall of air pressure inside of the bird's respiratory system. And here, what we're recording single units from is not one of these muscles larynx, but one of the respiratory muscles down in the trunk. So I won't go through this in detail, but analysis thus far suggests, just like we saw in cortex, that there seems to be a timing code at about the 1 millisecond time scale that's shaping behavior. And when I say shaping behavior, here I mean when the bird is singing, controlling things like vocal pitch. During breathing, I mean controlling air pressure. So okay, so that was the first thing that and I mean this is preliminary, but stay tuned. So this is some evidence that there is a spike timing code in the motor periphery. But remember, that's not all we need. We need to show that spike timing differences can actually affect behavior. The way we're going about this is as follows. So we are good at sticking tiny little wires in individual vocal muscles. So we can stick a pair of wires into the same muscle that I showed you on the previous slide. We can then, at low current and very precisely controlled times during song, introduce patterns of electrical pulses that give us a little bit of extra contraction of the muscle. So here, for example, what we're doing is we've got two different patterns of stimulation trains being delivered to this muscle. The numbers here show the interpulse interval. So we've got 3 milliseconds, 3 pulses, first two separated by 3 milliseconds, then 9 milliseconds. The blue is for 9-3. So the only difference between these stimulation patterns is that the middle pulse has been shifted over by 6 milliseconds. We can apply this during a particular song syllable while a bird is singing. And what this shows is the change in pitch from baseline when we're, and by baseline I mean a syllable in which we're not stimulating at all. So the change in pitch introduced by these two patterns is dramatically different, and this is a very consistent finding. So what this suggests to us then is that not only the muscles, but also the soft tissues that are being moved around are capable of transducing these millisecond scale differences into a difference in behavior. In collaboration with Koen Ellemann's lab in Denmark, his lab is running in vitro studies of the mechanical property, the force-generating properties of isolated muscle fibers. So what we can do is we can take bundles of muscle fibers out of this same muscle, put them in a dish, measure their force-producing properties. When we stimulate them with these same patterns, and summarizing quite a bit, we see similar, if not greater, differences in output. Just by shifting one of the stimulation pulses around by a few milliseconds. Koen, our collaborator, has also developed, and we've helped modify, this great prep where you euthanize the animal, you take the whole vocal organ out, put it in a dish, and the dish has a lot of tools in it that let you so we have complete control over the air pressure and air flows both inside and outside of the trachea. The muscles are all kept alive by being perfused with oxygenated saline, so the muscles still work. We can then blow air through the syrinx. The syrinx will actually produce sound. We can then control muscle contraction with electrical stimulation and film the whole thing with a high-speed camera at like 24,000 frames per second. So this is an image. This is a still frame from a movie. So here's this VS muscle. This is this muscle I keep talking about. This is the adjacent muscle to it. What this shows is that if we just specifically stimulate the VS muscle will shorten, the adjacent muscle will not, and we'll see these changes in the sound that's being produced by the preparation. So I'm out of time, but what I want to convey with this is that by combining, again, I think this points to one of the things that I really like about the songbird, is that by combining this really diverse range of techniques, in fact, this is a lot of different experimental techniques just to throw at this one question about spike timing for motor codes. And I hope this has been apparent from the other studies that we've done, is that by looking across at all these different relationships between these different levels of description, we can really learn interesting things about how the brain is coordinating these very complex skills and modifying them both based on sensory feedback. And I will stop there.
I know some folks probably have to run.
Yeah, go ahead.
So maybe in the interest of that time constraint, we'll take questions offline.